Well, hello, Gilpin County students. I figured I'd take a moment to at least introduce a little bit funnier this time since you requested it. But here is your first little mini lesson that has to do with something actually about content as opposed to me talking stupidly into the computer. I will try very hard not to say too much, but give me a break if I do. So this little piece here is about the cell cycle, something you've learned about already. So this should give you some practice on kind of taking notes or figuring out how you want to take your notes or how you can not watch other YouTube videos while you're trying to take notes. I said it. In terms of getting some information on here. So this little bit's about the cell cycle, mitosis specifically. So you've learned about this before. You can see some pictures on here. And hopefully, if you think about it, you should probably recognize what all of these different pictures are on here. We should remember interphase, prophase, metaphase, anaphase, telophase. Well, at least it's a way for us to take a look at it. Down at the bottom, I don't know, you didn't get a chance to see this because we were a little behind, but there's some actual pictures of what these cells would look like if we were to take pictures of them under the microscope. Those are all onion root tip cells that have been stained with different colors and things like that. So anyway, there are some pictures of what they look like. So let's go back and take a look at the next bit, what's going on in the cell cycle. So just as a review, what is the cell cycle? It is that sequence of growth, division stuff that a cell does. It's the cell's life cycle. It is what it does during its life. Could be anywhere from a day to years and years long. But all of these different things are things that cells have to do. So there's three main components to it. You can kind of see them up here. This one on the outside, all this yellow stuff, is what we call interphase. Interphase is the cell doing cell stuff. Those are when they do metabolism. It's when the cell's growing. It's when the cell's making all those things that they need to do. It's being an eyeball cell, being a muscle cell, doing whatever stuff it has to do. The one part of it that's a little bit different, this one in the middle here, we can't see that because it's yellow, is the synthesis phase right in the middle. So it's actually making some more of its DNAs to be able to move on into the next phase. The second part is called mitosis, and that's these little bits around the edges right here. And mitosis, you should remember, is about the way that the cell divides its nucleus, divides all that important chromosome DNA information into two equal copies. It's really important that you get all that stuff, otherwise you end up with cells that just don't work right. And then the last little bit is this kind of blue section up here at the very top called cytokinesis. And cytokinesis' job is to be able to divide up the rest of the cell. Not just the nuclear material, all that DNA, but things like mitochondria and Golgi apparatus and all kinds of other membranes and things like that. So the cells have all the parts that they need to do. So those are the three main parts of the cell cycle. Interphase, which is by far the longest. Mitosis, which is that nuclear division dividing up the DNA. And then cytokinesis, which goes along with mitosis. So in terms of doing this little bit of review on vocabulary, when we're talking about mitosis, you're talking about the movement of chromosomes. Chromosomes are that package of DNA. So here's a little diagram and some other information in here in terms of what is a chromosome, what the parts are. You remember what a chromosome is, that it's made up of two identical copies. Those two identical copies are right here. They're called chromatids or sister chromatids. They're linked together in the middle by something called a centromere. And that's a little kind of a protein-dense section. And the last part on here, and I know I'll go up and do the top part, genes are actually segments of DNA. There are little different regions on there. It shows one right here, but there could be a gene here, there could be a gene here, down at the end. There's genes up at the top. In fact, there's hundreds and thousands even of genes per chromosome. It's not just one. So a couple other things about chromosomes kind of interesting. You guys probably remember that human beings have 46 chromosomes. If you take a look at this cool little diagram, I kind of threw some stuff down here, bottom right. Sorry, my lovely square there. There's actually no relationship between the type of species or the complexity of a species, I guess, and the number of chromosomes you have. So yeah, a mosquito, pretty simple creature's got 6 chromosomes. Peas, as in the plant, 14 chromosomes. Yeast, 32. That's the stuff you'd make your bread with. Cats, 38. Humans, there's us, 46. Chimpanzees actually have 2 more than us, 48. A potato has more chromosomes than you do. Maybe that explains some things about a couple of you guys. I'm not putting any names on that. Dogs have almost double what we have, 78. And there's actually a particular one kind of species of fern, I didn't put the full scientific name on there, has over 200 chromosomes. So I don't think anyone would argue that a fern is more complex than you are. There may be 1 or 2 of you students that I could argue that. But in general, the number of chromosomes isn't really particularly related to that. Remember, chromosomes always come in pairs. You had to get a chromosome from your mom and a chromosome from your dad. One came in the egg, one came in the sperm, and that's how they came together to make you. So that's why we always have even numbers of chromosomes. And I think that's about all I need to do on this slide. So let's move along. All right, moving into the nitty-gritty stuff. What's actually going on in these individual phases? This is the stuff that you need to be able to demonstrate to me for proficiency. So if you haven't been listening, now is the time to get your brain going again and to be able to explain to me what those phases are, what kinds of activities are in there. So first of all, top left, we got interphase. Here's a nice little student drew this. Actually, it wasn't me, but I found it on Google just like you guys do. Nice little drawing of what an interphase cell looks like. You've got the DNA in the center and chromatin. It's all nice and relaxed, surrounded by a nuclear envelope. You can see the nucleolus in there. I bet you don't remember what the nucleolus does. Extra credit if you do. And then you've got the centrioles, which are, again, those little things that anchor the spindle fibers later on. Over on the left, I don't remember from that other diagram that I had, but there's actually a couple of different things that happen during interphase. Regular cell activity is called the G1 phase. DNA synthesis is in the S phase in the middle, and then organelles are duplicated, and it's also doing its life stuff during G2. But during this phase, remember, DNA is in chromatin, like that, and it looks all fuzzy like this. You can't actually see the individual chromosomes. It's in a relaxed form, so you can actually read it. One of the big things that distinguishes interphase from mitosis is this first step, is that these chromosomes become visible. And that's one of the first things that we notice. And that's actually how it was identified when the first microscopes were out there. All of a sudden, these chunky things started to appear. So this particular cell right over here has 4 total chromosomes, 2 short chromosomes and 2 long chromosomes. So those are 2 pairs. This would have a chromosome number of 4. What else happens? The nuclear envelope has to dissolve. You have to be able to let those chromosomes out and move around. The centrioles, again, the centrioles are these things up in here. They started off kind of next to each other, but in prophase they migrate to opposite ends. So there's one at the top and one down here at the bottom, opposite ends of the cell. And then these long spindle fibers, which are actually protein microtubules, part of the cytoskeleton, grow and start to kind of reach towards those chromosomes that are in the middle. And that's the end of prophase. Moving into metaphase. Metaphase is the middle phase. That's how I always remember what it is. These chromosomes line up on this center piece right across the center. Metaphase, they are in the center. How do they get there? It's the spindle fiber's job, actually. They attach the centromeres, that middle part of the chromosome, and kind of push and pull, sort of jostle those chromosomes into place and get them into the right place, and they get into line. Believe it or not, there's actually a type of a genetic checkpoint here that the cell can check to make sure that all of those chromosomes are lined up and all of them are attached before they move on to the next step. Because if there's a mistake here, that means that in the next phase, anaphase, where they move apart, that you may miss something. 2 chromosomes may go one way, and no chromosomes go the other way, and missing one in a future cell. So a little bit different. Anaphase, pretty quick phase. This is after they've lined up, after it's been checked, everything's in order, all of it's organized. The centromeres themselves, in other words, the middle part of the chromosome separates, and the spindle fibers contract or shorten. These things right along the lines, right? These get shorter like little bungee cords, and they pull those chromosomes towards the opposite ends. It's really important that those chromosomes start together and get pulled apart that direction so that you get one sister chromatid in each of the offspring cells. That's a really critical part. Last thing that happens, we didn't talk about this in class too much. The cell itself actually elongates. You can see this cell right here, kind of longer, more oblong than the metaphase one, which is pretty round. And again, why? It's to kind of help pull that cell into 2 different pieces. Because eventually it started round, it's going to have to become 2. So the actual spindle fibers kind of push that cell longer. In the picture it looks like this. But okay, moving on. Last bit, telophage. Check out that cleavage right there. Look at that. It's terrible. All you guys are having butt jokes as we're talking about it. Telophase. Telophase is that reverse of prophase. Those chromosomes start to relax, go back into that sort of non-visible chromatin form. If you take a look in the diagram over here, you'll see that those chromatids, are single. They're not double anymore, but there are 4. There's 1, and then 2, and then 3 goes around this way, and then 4 goes around that way. So you do have all 4 of them there. And they're kind of showing this one right here, starting to kind of unravel a little bit, relax into that bit. And then that nuclear envelope starting to kind of re-corral that DNA into that safe central zone. Then the last thing that goes right along hand in hand with telophase is cytokinesis. In cytokinesis, again, this is the movement of the cell into two separate parts. You've got to have that plasma membrane pinching in. That's this part right here, right? And you can see it on the side. We call that cleavage. Love it. If it's a plant cell, there's a cell wall there, so it can't really pinch in. So it actually just forms a new little cell wall chunk right across that center axis. And then it tries to make sure by of course, microtubules, microfibers, all those other little parts of the cytoskeleton, that you've got some of each kind of organelle in each cell. If you're missing mitochondria in the top cell, for example, that cell is not going to work. It's going to die. So you've got to make sure you have a little bit of everything. In the end, what's the whole point of this process? This is our last cell today, our last slide today. What should we get? It's critical, if it's done right, that you should get 2 and only 2 identical offspring cells. They're called daughter cells. I don't know why they picked that in terms of son cells or something else, but you should have 2 identical cells, same number of chromosomes that you started with, basically the same number of organelles, basically the same amount of cytoplasm. You've got to have all that stuff so that the cell can be functional. In single-celled organisms, like bacteria, this is actually how they reproduce. Kind of boring compared to what we get to do. But in those guys, it's called binary fission. That's the name of that process, where 1 bacteria becomes 2. I'm going to have to ignore my phone. Sorry about that. And those guys actually reproduce in this particular format. If you're a multicellular organism, like you guys, well, most of you are, mitosis is how cells or organisms replace their cells that they've got, repair ones that aren't working correctly. It's how the organism overall gets bigger. Remember, you started off as 1 single cell, and now you're trillions. You had to have cells reproduce to do this. And eventually, this also allows for this part right down here, which is actually a really important step. Having more cells allows cells to differentiate. You can have cells with different jobs. And it's kind of weird to think that you have 2 identical cells, but yet somehow one cell can become a muscle cell, and eventually another cell could become an eyeball cell. I'll let you kind of think about how that's even possible. They're identical. How can they have different jobs? So anyway, that's one of the results of mitosis. But they should have all started out with the same genetic information. Little not-so quiz right here on the side. Here's a picture of that same kind of onion root. Should be able to tell me which phase these all are. You've got phase A. You can kind of see these little squigglies down here. You've got phase B up in the top. See that little dashed line right in the middle? Phase C, there's your whole nucleus right there. You can kind of see the whole bit. Here's D. Just kind of fuzzy, and then here's E down at the bottom. So again, I know the answers are right down below. I know you've already looked at it. But you can tell that this one's in anaphase. Why? Those chromosomes are in that moving away phase from one another. In cell B up here, here's that new cell plate. This is actually telophase, right? That last almost 2 new daughter cells, it's in that process right there. This is interphase. This is what a normal cell nucleus looks like. You can see that DNA in there, that chromatin just kind of looks sandy. This darker purple circle in the middle, that one right there, is the nucleolus. But that's what an interphase, regular normal cell nucleus looks like. This over here, on the other hand, is what it looks like in prophase. You can kind of see those chromosomes are starting to get a little bit chunkier. It's not quite as distinct as the interphase nucleus is over there. And then the last one right here, all these chromosomes are kind of in a line. These are kind of some of the long hanging pieces that are kind of hanging in there. They're mostly tried to pack in that center, but some of them are overlapping. So that's a metaphase cell. It kind of looks like a mustache right in the middle. So there you have it. That's what mitosis is: cell cycle, interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. See you in class tomorrow, slackers. Quit looking at YouTube.